We hit glioblastoma with radiation that's designed to literally shatter its DNA.
Right. It's the most aggressive primary brain cancer there is.
Exactly. And this a standard clinical algorithm deployed against it is, I mean, it's brutal, right? You have surgery to cut it out, radiation to burn what's left, and then a heavy chemotherapy drug called temozolomide to just poison the remnants.
Yeah, you would really think that overwhelming arsenal would be enough.
You would. So why does the tumor use that exact treatment to adapt, survive, and almost inevitably come back stronger?
Yeah, I mean, it's kind of the ultimate paradox in neuro-oncology. We throw our most destructive tools at this mass of cells agents that physically break the double helix of its genetic code,
Right.
and yet it finds a way to not just survive the assault, but to actually rebuild its armor and keep growing.
And that paradox is exactly what we're tackling today for you guys.
Yeah.
We've got our hands on a really groundbreaking 2026 paper from Cell Death and Disease.
It's a fascinating read.
It really is.
Yeah.
So the mission for this deep dive is to explore a totally novel approach to this devastating problem. We're going to unpack how starving a brain tumor of one single specific nutrient and amino acid called arginine can, well, fundamentally rewrite its DNA defenses.
Making it completely helplessly vulnerable to that same radiation.
Okay, let's unpack this. If we want to understand how this new treatment works, we first have to understand the enemy's primary defense mechanism, right?
Right, you have to know how it defends itself first.
So when that radiation hits the brain and shatters the tumor's DNA, what actually happens inside that cancer cell? Like, does it just have microscopic machinery waiting around to stitch itself back together?
Essentially, yes. It has this hyperactive system called DNA damage repair, or DDR.
Okay.
When radiation passes through a cell, it causes severe structural damage. We're talking single-strand breaks and double-strand breaks right in the DNA backbone.
And to survive that, a glioblastoma, or GBM, just activates these highly specialized responder pathways.
Exactly. They use pathways like base excision repair, homologous recombination, and non-homologous end joining. But to coordinate all that, there are two key proteins pulling the strings.
And the paper focuses heavily on these two, right? PARP1 and LIG1. What exactly are they doing?
So think of them as the emergency crew. PARP1 is the cellular alarm system. It's the very first responder.
So the moment a strand of DNA breaks,
PARP1 senses the damage, binds to the broken site, and chemically signals the rest of the repair crew to rush to the scene.
wow. And what about LIG1?
LIG1 is the ultimate molecular glue. Once the surrounding machinery has rebuilt those missing genetic letters, LIG1 swooped in and chemically seals the repaired DNA strands back together.
And in GBM, these proteins are heavily upregulated, right? The tumor is simply repairing itself faster than the radiation can destroy.
Exactly. It's just out-repairing the damage. But this seemingly invincible repair system has a crack in the armor.
Okay, and this is where it gets interesting. It all hinges on an enzyme called ASS1.
Right. Arginosuccinate synthetase 1. It's a critical enzyme required for a cell to synthesize its own arginine.
Which is an amino acid.
Yeah. Healthy cells can produce this enzyme and make their own arginine if they run low. But the researchers note a major vulnerability here.
Because
About 30% of glioblastomas are ASS1 negative. They have genetically lost the ability to produce this enzyme entirely.
meaning they absolutely cannot make their own arginine. Biologists call them arginine oxotrophs.
Yes,
Right.
arginine oxotrophs. They are entirely dependent on scavenging it from their environment, basically stealing it from the patient's bloodstream just to survive.
And that leads to a really fascinating paradox when you look at the clinical data.
Yeah.
The paper mapped the RNA sequencing of 23 primary GBM patients from the CGGA database.
And what they found was wild. The patients with ASS1 negative tumors, the ones who couldn't even make their own basic nutrients,
Yeah.
they actually had much higher DNA repair capacities and significantly worse overall survival rates.
Wait, hold on. I need to challenge that because it makes, like, no biological sense to me.
That sounds backward, I know.
Right. If you are this hyper-aggressive cancer, practically invincible against radiation, why on earth would you evolve a fatal flaw where you lose the ability to feed yourself? It seems like a massive evolutionary step backward.
It definitely seems counterintuitive until you think about biological trade-offs. Think of the tumor as a hyper-aggressive invading army.
Okay, an army.
If that army wants to march at lightning speed and dedicate absolutely all of its energy to weapons and armor.
Which in this case is rapid cell division and heavy DNA repair.
Exactly. If it does that, it can't carry a bunch of heavy farming equipment. So it deliberately burns down its own agricultural supply lines, the ASS1 enzyme.
Oh, wow. It sacrifices self-sufficiency for raw speed.
Yes, assuming it can just pillage all the food it needs from the local environment as it expands.
Okay, let me pause right here and do a quick 5-minute recap for everyone listening to make sure we've got this foundation solid. Good idea. So, glioblastoma survives our best weapons by hyperactivating DNA repair proteins, specifically the alarm, PARP1, and the glue, LIG1.
Right.
But to achieve that aggressive state, 30% of these tumors traded away their ability to make their own arginine. So we have this terrifyingly strong armored army, but it's entirely dependent on outside rations.
That's the perfect way to look at it.
So for you listening, if you know the enemy has no rations and has to have food delivered to the battlefield, how do you defeat them? You just intercept the delivery, right?
That is exactly the strategy. You cut off the supply chain, and the weapon they use to do that is a specialized drug called ADI-PEG20.
ADI-PEG20.
Correct,
It's an engineered enzyme, right? What exactly is its mission when it enters the body?
Its sole function is to circulate in the bloodstream and systematically degrade all the extracellular arginine it bumps into.
Just wiping it out.
Yeah, it breaks the arginine down into citrulline and ammonia. It essentially functions as a molecular vacuum, sweeping the blood completely clean of the tumor's only food source.
But here's the part that I found so incredibly elegant. When we talk about treating brain cancer, the biggest hurdle is, always the blood-brain barrier.
Oh, absolutely. The BBB is this highly selective fortress that stops almost all chemotherapy drugs from ever reaching the brain.
But ADI-PEG20 bypasses that problem entirely.
Because it doesn't even need to cross the barrier.
Wait, it doesn't enter the brain at all?
It never has to. It just stays in the body's main circulatory system, cleaning the blood outside the brain. But because it creates a systemic vacuum, it drains the arginine from the brain's local environment anyway.
That is so smart. The tumor is locked inside the skull, trying to order takeout, and suddenly realizes the supply lines have completely dried up.
Exactly. So the researchers tested this on patient-derived cells in the lab. They compared ASS1 negative cells, specifically lines called GBM96 and GBM31, against tumors that could still make their own arginine, like the TB48 line.
And what happened when they treated those cells with just the ADI-PEG20 drug by itself?
Well, the growth of the negative cells significantly slowed down. They were really struggling. Meanwhile, the positive cells were completely unaffected because they just kept making their own food.
But slowing growth isn't a cure.
No, it's not. The real magic happened when they combined this starvation tactic with radiation.
Let's talk about how they measured that, because the timeline here is super critical. When radiation breaks a double strand of DNA, how do the scientists actually see that damage in real time?
They look for a highly specific molecular flag called phosphorylated gamma H2AX.
Gamma H2AX.
Right. When a DNA strand snaps in half, a histone protein that sits right next to the break immediately gets phosphorylated. It's basically the cell throwing up a bright red flare saying, "We have catastrophic structural damage here."
So by measuring those flares, researchers can quantify exactly how badly the DNA is shattered.
Precisely, and the timeline they discovered is just staggering.
Yeah, I was amazed by this.
When the dependent cells were starved with ADI-PEG20 and then hit with radiation, those gamma H2AX flares spiked incredibly fast. The amplified damage was visible within 30 minutes.
30 minutes. And it peaked at exactly 1 hour post-radiation.
Yes. It's like hitting a fighter with a massive left hook precisely at the split second you've tied their hands behind their back.
They can't put their guard up, and the damage is immediate.
And what's fascinating here is how violently that translated when they moved from petri dishes to living systems.
Oh, right, the mouse models.
Yeah, they used patient-derived xenograft or PDX mouse models where they implanted human glioblastoma cells into the brains of the mice.
And when they subjected those mice to both the ADI-PEG20 starvation and a 4 GI dose of radiation, the tumors didn't just stop growing, right?
No, the physical volume massively shrank.
And if you look at those shrinking tumors under a microscope, how violent is this death? Is the cell just quietly powering down, or is it something more dramatic?
Oh, it's total biological devastation. Pathologists use 3 very distinct terms for what was happening here. First is nuclear pinosis.
Prognosis, what does that look like?
That's where the core nucleus of the cell irreversibly shrinks and condenses into a dense, dark mass. Think of it like a grape rapidly shriveling into a hard raisin.
Yikes.
Then that's followed by karyorrhexis, where that shriveled nucleus literally shatters in fragments, blowing pieces of genetic material all over the inside of the cell.
It just explodes.
Basically. And finally, karyolysis, which is the complete dissolution of those fragments as enzymes just melt them away. The tumor cells were quite literally disintegrating.
Okay, let's do another quick recap of this section for you guys listening, just to map the timeline of this attack.
Yeah.
So we have a drug that creates a systemic vacuum of arginine in the blood, starving the tumor without ever crossing into the brain.
Right.
When we follow that starvation with radiation, the tumor's DNA shatters, peaking exactly 1 hour after the blast. And that leads to total cellular disintegration.
Exactly. So mentally flag that 1 hour peak time, because if doctors know the exact 60-minute window when a tumor is completely defenseless, that could fundamentally change how we time radiation doses in a real-world clinical algorithm.
Right. It takes it from a guessing game to precision timing. But there is a massive logical gap here that we have to cross.
The how?
Yes, we know the drug works. We know it starves the cell, and the cell dies violently when irradiated. But how? How does removing an amino acid floating around outside the cell suddenly reach deep into the nucleus and stop DNA repair proteins like PARP1 and LIG1 from doing their jobs?
To answer that, we really have to go deep into the molecular engine room. We are looking at the intersection of metabolomics and epigenetics here.
Okay, I'm ready.
So the researchers needed to see what the genes were doing, so they profiled the tumor's transcriptome using microarray data from the GBM31 cells.
A transcriptome is basically the sum of all the active blueprints the cell is reading, right?
Yeah, exactly. And they found that 24 hours after the tumor was starved of arginine, 183 genes were significantly downregulated. And they weren't random.
They were heavily clustered around DNA damage repair. The specific blueprints for PARP1 and LIG1 were completely silenced.
Yes. The cell wasn't just struggling to repair its DNA. It had literally stopped manufacturing the tools to do the job.
Here's where it gets really interesting. Why did those specific blueprints get locked away? To find out, they mapped the cell's metabolome using liquid chromatography mass spectrometry.
LCMS, right. The metabolome is the complete chemical soup of small molecules inside the cell. And they looked at it at exactly the 6-hour mark, long before the genes shut down at 24 hours.
Right, capturing a snapshot of the chemical metabolites. And what did they see at 6 hours?
They saw a massive disruption in a network called one-carbon metabolism. This is a complex cycle, primarily the folate and methionine cycles, that cells use to pass around single carbon atoms for essential functions.
Walk me through that disruption. What exactly changed in that chemical soup?
Well, several metabolites like choline, betaine, and S-adenosylhomocysteine, or SAH, plummeted. They decreased significantly.
Okay, but what increased?
Glycine went up, but the real smoking gun was a massive spike in a molecule called SAM, or S-adenosylmethionine.
SAM?
Yes. And the researchers looked at the SAM to cell-H ratio, which is the ultimate index of a cell's methylation potential.
Mm-hmm.
Because of the spike in SAM and the drop in LSAH, that ratio skyrocketed.
And SAM is the universal methyl donor in a cell, right?
Mm-hmm.
If a cell needs to add a methyl group, which is just a carbon atom bonded to 3 hydrogens onto a protein or onto its DNA, it takes it from SAM.
Exactly.
So the starvation triggers this metabolic panic, and suddenly the cell has a massive excess of this methyl donor SAM. But what does this cell actually do with all of that? How does that chemical imbalance turn off a gene?
That brings us to epigenetics. Genetics is the hardwired sequence of your DNA. Epigenetics refers to the chemical tags that turn those genes on or off without changing the sequence, like a physical volume knob on a stereo.
Right.
Because the starved cell was flooded with SAM, it started aggressively over-methylating its histones. Histones are the spool-like proteins that your DNA wraps around to stay organized.
And the researchers ran chromatin immunoprecipitation assays, ChIP assays, and isolated one very specific epigenetic tag that was just going crazy, right?
Yeah.
H3K27E3.
Yes, H3K27E3. It's a highly repressive tag. When this chemical mark is placed near a gene, it causes the DNA to wind up incredibly tight, like a knotted shoelace.
So the cellular machinery can't read it anymore.
It is the ultimate silencer. And the GAP assays physically proved that after the arginine starvation, there was a massive accumulation of these H3K27E3 tags binding directly to the promoters of the PARP1 and LIG1 genes.
Okay, I love the analogy you used for this when we were prepping.
Oh, the red paint.
Yes, walk everyone through that because it makes it so clear.
Right, so to visualize this, think of that massive spike in SAM as a giant bucket of red paint. And the H3K27E3 tag is a giant stamp that says, do not transcribe. When the cell is starved of arginine, the metabolic engine misfires and accidentally knocks over the bucket of red paint. The nucleus is flooded with it.
And in the ensuing chemical chaos,
the cell accidentally stamps do not transcribe right over its own critical blueprints for DNA repair.
That is mind-blowing. It really is. But in science, correlation isn't causation.
Right.
Just because the paint is there doesn't mean it's what caused the cell to die.
Mm-hmm.
How did they prove that the epigenetic silencing was the direct cause of the vulnerability?
They ran rescue experiments. They basically gave the tumor a way out to see if it would survive. In one experiment, they used a drug called GSK126, which is an H3K27E3 inhibitor. It specifically blocks the enzyme responsible for making that repressive stamp.
Blocking the stamp. And what else did they try?
In another experiment, they used lentiviral vectors to forcibly overexpress PARP1 and LIG1, artificially bypassing the locked blueprints so the cell kept manufacturing them.
And the result of those rescue experiments?
The radiosensitizing effect of ADI-PEG20 completely vanished. Yeah, when they blocked the red paint, or when they forced the tools to be made anyway, the tumor regained its armor and easily survived the radiation, even while starving.
The mechanism is airtight.
The starvation causes the epigenetic silencing. Period.
Okay, let's track the full chain reaction for you guys, from outside the cell to inside the nucleus, because it completely reframes the disease.
We deprive the blood of arginine.
The tumor, unable to make its own, goes into a metabolic crisis, leading to a massive spike in SAM.
That excess SAM acts as red paint, driving a huge increase in the repressive H3K27E3 epigenetic tag.
Those tags physically bind to and lock away the blueprints for PRP1 and LIG1.
We hit it with radiation, the DNA shatters, there are no tools to fix it, and the cell disintegrates.
It's an incredible cascade.
It really is. And for you listening, does this fundamentally change how you view cancer? Not just as a disease of mutated broken DNA, but as a deeply metabolic machine where the food it consumes literally controls the structure of its genetics.
It absolutely changes the paradigm. It forces us to look at cancer as an entire ecosystem. And it highlights exactly why metabolic therapies are becoming the true frontier of modern oncology.
So what does this all mean for the immediate future of treatment?
Well, if we zoom out to the clinical landscape, this mechanism solves a massive, expensive frustration in pharmacology.
How's that?
Right now, billions of dollars are being poured into developing PRP inhibitor drugs for clinical trials. The goal is to do exactly what we just discussed. Block the DNA repair alarm so radiation works better.
But they hit a wall, right?
A literal wall. Those traditional physical drugs struggle immensely to cross the blood-brain barrier. They just can't get into the brain in high enough concentrations to be effective as radiosensitizers.
But this approach sidesteps the fortress completely.
Right. It leverages a concept called synthetic lethality.
Synthetic lethality, explain that.
It's a scenario where a cell can survive losing one function, but if it loses two specific functions at the same time, it dies. By using a systemic metabolic sweep cleaning the blood with ADI-PEG20, we are remotely triggering the simultaneous inhibition of both PRP1 and LIG1 inside the brain.
We bypass the blood-brain barrier entirely by forcing the tumor's own metabolic panic to act as the drug.
It's brilliant.
It's the ultimate Trojan horse strategy. You don't assault the heavily fortified castle walls with traditional drugs. You just dam the river flowing into the city and wait for the starving inhabitants to lower their own gates.
Exactly. And this raises critical questions for clinical trials moving forward. Now that we have the proof of concept that metabolic manipulation can profoundly sensitize these aggressive tumors, the next phase is optimization.
Like timing the doses.
Yes. How do we take this highly synchronized 60-minute window of vulnerability and reliably replicate it in a human patient receiving daily radiation?
Which brings us back to that very first question about the blunt brute force algorithm we currently use to treat glioblastoma. The standard treatment is incredibly powerful, but it is entirely blind to the metabolic and epigenetic shifting sands happening inside the tumor in real time.
It's a static treatment for a highly dynamic disease.
Right. But what if the future of oncology isn't just about finding a bigger hammer? I want to leave you all with a final thought to mull over or explore on your own.
Ooh, this is a good one.
If simply altering a tumor's diet by temporarily removing a single basic amino acid from the bloodstream can literally rewrite its epigenetics and shut down its deepest defenses, what other metabolic levers exist in the human body?
There have to be more.
Right. Could the future of cancer treatment involve precisely altering your entire systemic metabolism, perhaps adhering to a highly specific fast, or taking an enzyme that cleanses your blood of a particular nutrient for just a few hours before you walk into a radiation clinic? Could we turn the very food a tumor aggressively craves into the exact key that unlocks its destruction?
It's a profound perspective shift. We're no longer just trying to poison the tumor. We are actively manipulating the environment it desperately relies on to survive.
It is a brave new world of precision medicine. Thanks for joining us on this deep dive. Stay curious.